Sardi here. I am going to give you what presumably will be your first lecture for Unit 3. If I can get my pen up here. Unit 3 will cover the human reproductive system, and so as part of that process you're going to be learning about how sperm cells from males and egg cells from females come together at fertilization to form the first cell of a new human individual. And there's a critical process that's necessary to make that happen called meiosis. So that occurs in both males and females. So I am going to provide an overview of that process to you as part of this lecture. I'm sorry I don't have the camera on today. I don't have access to the computer where the camera is located. And I know you're absolutely crushed that you can't see my shining forehead there on the screen as we go through this lecture. Before we talk about this meiosis process, we need to do a quick overview again of chromosomes and genes and what those are. The cells that make up your body are often referred to as somatic cells. Well, I won't say often, but you may run across that term in your textbook. Somatic cells are your typical body cells, what you typically think of as a cell. Your red blood cells are an exception. Your red blood cells punt out their chromosomes before they mature. But otherwise, your somatic cells have 46 chromosomes in them, as you've learned about before. And those 46 include 2 copies of chromosomes number 1 through 22, and then you have your 2 sex chromosomes. Those are present in almost every somatic cell in your body. Sex chromosomes, you've either got 2 X chromosomes, which makes you a female, and if you have an X and a Y chromosome, that makes you a male. And as you've learned about before, chromosomes are very large DNA molecules. DNA molecules are chains of A, G, and C and T nucleotides, like you've learned about before, and 2 of those chains or strands are bonded together and twisted to form this double helix shaped molecule that you see here. Kind of looks like a spiral staircase. So chromosomes are very large DNA molecules, and they're complexed with various proteins to help wad them up and cram them into the tiny little nuclei in each of your cells. Now along your chromosomes, those big DNA molecules, at particular locations, you have stretches of A's and G's and C's and T's in particular orders or sequences that function as genes. And genes are blueprints or recipes for the thousands of different types of proteins that the cells in your body need to make. And proteins, as you've learned, are the workhorses of your cells. They indirectly or directly lead to all of the different traits and functions of your body. So genes are, those blueprints are very critical, which means all of the cells of your body, other than those pesky red blood cells, have got to contain 46 chromosomes. They've got to have 2 copies of chromosome 1 through 22 and the 2 sex chromosomes. Otherwise, they don't have a full set of functioning blueprints or recipes for all of those critical proteins that those cells need to make. And also, you guys have learned before, and this is where sexual reproduction is going to come in, 2 copies of chromosome 1 through 22. Well, guess what? You get 1 copy of each of those from mommy and 1 copy from daddy. And then you get, from mommy, you get 1 X chromosome, and then from daddy, you either get an X or a Y chromosome. And that combination from your parents determines whether you're a female or a male. And again, guys, you've got to have the proper combinations of these chromosomes in a cell, otherwise that cell is going to die or it's going to function very inappropriately. All right, so sexual reproduction, which is the topic for Unit 3, you guys already know that requires that a sperm cell from a male must fuse with an ovum from the female. That's ovum is another name for what we more commonly call an egg cell. When those two come together, like you see over here in the picture, that forms a zygote. That zygote is the first cell of the new embryo that's going to form, and which will, of course, lead to the new human individual. Now, that zygote is that single cell is going to multiply over and over and over again through the mitosis process that you've heard about before. And so that zygote has got to have 46 chromosomes in total. And as that zygote divides into two daughter cells, each of those two daughter cells will have those 46 chromosomes. And as each of those daughter cells divides, the daughter cells of both of those will have 46 chromosomes, and so on as you wind up creating the trillions of cells in the body that make up the new human. So we have kind of a problem here. Got kind of a problem. Sperm cell is going to fuse with an egg cell, an ovum. So you can't have 46 chromosomes in a sperm cell and 46 chromosomes in an ovum, because that would give you 92 chromosomes in the zygote. So therefore, we need sperm cells to have only 23 chromosomes, and we need ova to only have 23 chromosomes, so that when they fuse together, you wind up with 46 in the zygote. By the way, guys, you will see this term gametes. Gametes is a term that refers to the special cells that are used for sexual reproduction, your sperm cells and your ova, that only contain 23 chromosomes. Okay, so meiosis is the process that is concerned with generating these cells that instead of 46 chromosomes apiece, only have 23. That also means that, if I can draw it in here, instead of having 2 copies of each chromosome, they're only going to have 1 copy of each chromosome, and that is sometimes designated as 2N versus N. 2 is referring to the number of chromosomes, or I'm sorry, the number of copies of each chromosome, and N is referring to the number of chromosomes itself. So 46 is 2N. Another term for that is diploid. A cell that contains 46 chromosomes in a human is diploid. A cell with 23 chromosomes can be referred to as haploid, and you'll see it as N for haploid and 2N for diploid on many of the diagrams in your textbook. So this is what we're doing in meiosis. This contrasts with the mitosis process that you have learned a little bit about before, that you see over here on the left-hand side of this diagram. Mitosis, you're going from a mother cell with 46 chromosomes, that's going to divide, and you're going to have 2 daughter cells, and each of those is diploid with 46 chromosomes. During meiosis, the process that occurs, I'm going to erase. During meiosis, you're going to start with a mother cell that has 46 chromosomes. And you're going to end up with, there's 2 phases, but at the very end of the process, you're going to end up with 4 daughter cells, each of which has 23 chromosomes at the end. So this is meiosis over here on this left-hand side of the slide, or the diagram. Meiosis is divided up into 2 phases. And what they're doing here on the diagram, of course, is they're not going to draw in 46 chromosomes, because that would take up the whole page to show all of those. So what they're showing you here are 2 copies. You got two copies of a pink chromosome. Let's say that's chromosome number 5. and 2 copies of a blue chromosome, we could say that's chromosome number 14. Each of those copies has been duplicated or replicated, just like what happens during mitosis. So that's why they look like H's, because we're saying we got 2 pinks and 2 blues, but each of those pinks and blues has been duplicated. That's why they look like H's or X's at this point. So unlike what happens during mitosis, where you're going to wind up with 2 daughter cells, each of which will contain 46 chromosomes, we're first going to do a process called meiosis 1, where the daughter cells will only get 1 copy of each chromosome. Okay, so you end up here with 2 daughter cells that are N, or haploid. So up here in the mother cell here, let's say you had 2 copies of 5. I'm sorry, this would be the other 5 here. And 2 copies of 14. Down in here, each of these just has 1 copy of 5, 1 copy of 14 in those daughter cells. Now those copies are still duplicated. They're in a duplicated state. So the next phase, which is called meiosis 2, each of these daughter cells from meiosis 1 will divide, and those duplicated copies get split and sent to different daughter cells. So at the end of meiosis 2, you wind up with 4 daughter cells containing 23 chromosomes apiece in a non-duplicated state. So they no longer look like X's or H's. All right, so that's an overview of what has to happen during meiosis. Where does this occur in males and females? Now, the male process of meiosis is referred to as spermatogenesis, and the female process is referred to as oogenesis. So it's oogenesis and not oogenesis. Spermatogenesis occurs in the testes in males, and more specifically within the testes, this is occurring within the walls of the seminiferous tubules. And you'll be learning more about that in future lectures. You'll also be taking a look at seminiferous tubules under the microscope in lab. Oogenesis occurs primarily in the ovaries, but it actually finishes in a fallopian tube at the time of fertilization. So just a little quick overview of what's going on in a male. Now you're going to hear more details about this story when you more specifically cover what happens in a male. But there is a cell, a diploid, or there are cells, diploid cells in these seminiferous tubules in the testes of males called primary spermatocytes. Those are the diploid mother cells. So those cells divide and they undergo the meiosis 1 process and lead to 2 haploid cells with 23 chromosomes apiece. And those are called your secondary spermatocytes. Each of those 2 secondary spermatocytes will divide through the process called meiosis 2, and that leads to 4 haploid daughter cells, each of which contains 23 chromosomes. And now those chromosomes are no longer in that duplicated or replicated state. These cells at the end of meiosis 2 in males are called spermatids. They don't yet look like, they don't have the morphology or the appearance of what we think of as a sperm cell as having. So the rest of, as you'll learn later, the rest of the maturation process in these seminiferous tubules involves these guys maturing into what you think of as sperm, the cells with the long tails on them that allow them to swim to the oocyte in the uterine tube and fertilize that oocyte. All right, so let's take a little bit more of a look at what's going on in the female. And you're going to have a lot more details again with the female in future lectures. But in females, again, the process of meiosis is called oogenesis, and it starts with cells that are called primary oocytes. In the ovaries. So these guys are in the ovaries. Each of those 46 chromosomes is in a duplicated state, like we looked at on the last slide. This cell divides into two cells. It undergoes the meiosis I process, each of which contains 23 chromosomes in a duplicated state. One of those cells, now this is very different from what we just saw with spermatogenesis. One of those cells is officially the secondary oocyte that's going to continue with developing the mature egg cell or ovum. The other daughter cell is called a polar body. The polar body is going to divide, so it is going to go through the next meiosis phase as well. But that polar body is non-functional. It's just going to die and be absorbed by the mother's body. So this is a non-functional daughter cell that's generated after meiosis I. Meiosis II, again, that's the process where these duplicated 23 chromosomes have to be split up among daughter cells. So the secondary oocyte will undergo meiosis II, and that's going to very briefly generate the mature egg cell that's called an ovum. The other cell, the other daughter cell from meiosis II, is going to become another polar body that you see here. So that's going to be non-functional. Has the chromosomes in it, but it's not going to do anything. And then again, your original polar body up here, it's actually going to undergo meiosis II and generate two more polar bodies. So at the end of oogenesis, you have one ovum plus 3 polar bodies. That's in contrast to what you see over here with spermatogenesis, where you start for every primary spermatocyte you start off with, you wind up with 4 haploid spermatids, each of which has 23 chromosomes, and each of those spermatids can be functional. Okay, and then as you see here, last part of the diagram, here comes the sperm cell with its 23 chromosomes, and you got your ovum here with mom's 23 chromosomes, and those two are going to fuse their chromosomes together and create the first cell of the new human individual, the zygote, which is going to have 46 chromosomes, 2 copies of 1 through 22, a copy each from mom and a copy of each from dad, and then plus either the 2 X chromosomes if this zygote is going to become a female, and an X and a Y chromosome if it's going to become a male. All right, so that's going to conclude our overview of the meiosis process, and again, you'll be hearing more of the nitty-gritty details about what's going on in males and females in future lectures. So thank you very much.